If I say the word protein, what does that make you think of? A lot of people would probably answer meat, but here's what you might not have thought of. We are all meat, and proteins are everywhere in us. In fact, they're responsible for all of our bodily functions, and they are divided into many categories that you might have heard of. For example, hormones, those are proteins, enzymes are proteins, antibodies, they're also proteins. And as such, it is very important to understand what these different proteins look like, so their structure, and how they are affected by different drugs. However, proteins are simply too small to look at using a regular microscope that uses light. Indeed, the wavelength of visible light itself is just too big to see any details. So instead, we look at the structure of protein using a cryoelectron microscope, which uses electrons, the parts that are orbiting around the atoms, since these have a much shorter wavelength. The electron beam that passes through the protein sample gives us many 2D images of the protein from all sides, which then allows us to reconstitute a 3D image of the structure with the help of powerful computers. The protein that I am studying is called melanocortin 3 receptor, or MC3, and in humans, it has been shown to affect weight, childhood growth, and the timing of puberty. So far, only one structure of this protein has been published, but I'm trying to get a structure of what MC3 looks like when it is in contact with a particular drug called Imcivree, of the scientific name setmelanotide. This drug is used to treat a very similar related protein, the melanocortin 4 receptor, but we know that it also interacts with MC3. This is why it's important to be able to see how setmelanotide might affect the structure of MC3 to understand, for example, what makes it effective and if you should expect side effects from the use of this drug.